structure that is genetically identical to its ancestor is a natural process that is present in nature. It occurs in the asexual reproduction of living beings, such as bacteria, plants, and even identical twins. Early in development of an animal, the embryo splits in two. There are two ways to make an exact genetic copy of an organism in a lab, and one is the artificial embryo twining, and the other one is the somatic cell nuclear transfer. This experiment showed that each cell in the early embryo of a sea urchin has its own complete set of genetic instruction and can grow into a full organism. Once separated, each cell grows into a complete sea urchin.
The scientists used a small electrical shock to separate one cell from 8-cell lamb embryo. The new cell started divided, and after a few days they placed the lamb embryos into a surrogate mothership. The result was the birth of 3 live lambs. This experiment showed that it was possible to clone a mammal by nuclear. The scientists created a lamb by transferring the nucleus from an adult sheep's udder cell into an enucleated egg. Never before had a mammal been cloned from an adult somatic cell. When an adult cell nucleus is used as a donor, its genetic information must be reset to an embryonic state. After some experiences, only one produced an embryo that was carried to term in a surrogate mother. This famous lamb was named Dolly. Her arrival started conversations about the implications of cloning. After the successes leading up to Dolly and Polly, several more animals had been successfully cloned.
Some scientists used somatic cell nuclear transfer to create a human embryo that could be used as a source of embryonic stem cells. The resulting stem cells lines were specific to the patient they came from, a baby with a rare genetic disorder. In this experiment, researchers took a skin cell from the patient and fused it with a donor egg cell after they stimulate the egg to being divided.
And you, what is your opinion?